Okay, so let's getting started. Good morning, everyone. Welcome to the session Translational Biology. So reminding you, this is the last session of the HDR workshop for knowledge-guided machine learning, which is part of NSF HDR program. So this is the second year of the funding, and so we are in the last session on the translational biology. So hope to remind everybody to submit your question through the Slido channel. Okay, so today in this session we have 5 distinguished speakers, and before we get to their speech, I'd like to make a short introduction in the session. So our focus is on knowledge-guided machine learning, in particular, how the knowledge can be transferred in the translational biology domain. And so in this, I hope to mention the one of the main issues in applying AI machine learning approach in the biology is the big P small n problem. So as you all know, so the deep learning has been popularly used everywhere, including the biology domain. And it has been especially the very famous multi-omics data, how to integrate multi-omics data for different disease predictions. But we are facing the huge problem is the small samples, but very large dimension and features. And some of the features may not be complete. So that I bring you the famous TCGA data sets. You can see they have 33 different cancer types with 20,000 primary cancer samples. However, if you look into different types, each type of cancer only has a few hundred samples. And the best we have is a little bit over 1,000. So definitely this is each type is not sufficient, the number of samples to be used to train a deep learning model. So that's the problem we are facing, how to apply machine learning effectively in small samples. So with that, I hope to introduce the latest machine learning community. So with the machine learning community also realized this problem for big data and small, long tail for small data problem. So a lot of research have been, working on this few shot learning problems. So with that, we have, so the biggest problem we have here is how do we see just a few patients and with different diseases, and then giving you a new patient, we can detect whether a new patient has what disease. So it's called a few shot learning. And so with that, what we generally idea is to transfer knowledges. So what we can do is to look back how humans do this. They transfer effectively any knowledge they can to the new events or new things. So for example, even a young kid can learn pretty fast by looking at a few samples. So with that, transfer learning has been very effectively used in the biology domain. And the latest in machine learning field is called meta learning, which uses the knowledge learned and to relevant tasks. So with that, I hope to mention the two famous approaches in this for the transfer learning is basically original transfer learning, which you train. There are different kinds of knowledge to be transferred. The most popular one is to train the neural network model with some related tasks and transfer the parameters to target tasks. So that's what the original transfer learning is. And now the latest meta learning is proved to outperform the regular transfer learning. Basically, here you are training the model with many small tasks, and these tasks including the support set and query set, so you can quickly from the support to the query with many different tasks. And supposedly, once you train your model can quickly adapt to new tasks, then you can use this parameter you trained and with a small sample as a support to adapt the model and get the new prediction model. And so that's what people have been proposing and applying to the bioinformatics domain. And with that, I bring back this TCGA data set, and you can see they are small samples, but they have a huge amount of features. And then we actually, I want to show you some of the results we have done to show how meta learning can be effective in applying to this kind of data. And basically, you have all different samples with different cancers, and with small tasks with each cancer, a few samples and prediction, and then you train the model and adapt it to the new model. And I will skip that. So basically, I want to show you the result on this. So basically, we train the model with meta learning from 31 cancers because there are 2 cancers are too small and the data are not adequate. So and then we validated and we tested in the way. So here are some of the results we did in the TCG data sets with proteomics data features as features alone, and using more than 7,000 patients over 33 cancers. And we compared with the traditional KNN random forest method and some of the meta learning method. And this particular meta learning method called MEMO came to be the best when applying to 3-way means 3 classes you can detect and 5 classes you can detect. So in that you can see the meta learning can transfer better in detections. And so with different features, you know, the TCGA has RNA feature also, that the result came similar. And so using all the features, actually the result is similar, but slightly lower because there are noises. So that gives us encouragement to apply meta learning to small sample environment for predictions and transfer learning. So we actually compared substantially between the transfer learning and the meta learning and see how they behave, how they do better. So I want to show you the result when we compare. So we trained the model using 12 different cancers, and then transfer learning. This is the transfer learning first, And this time we did not use the prediction, we used the survival and death event as a prediction. And then we use 4 new type of cancer, and then see how transfer learning and new meta-learning memo perform between them. So you can see mostly the meta-learning will transform the knowledge much better than transfer learning. But there are some cases transfer learning does better, and with the data probably more consistent between the new cancer and the 12 cancer we compared. So there are some, but you know, the performance is not far away between those. So these are the performance between transfer. So I think we can claim the meta-learning can do better than transfer learning in transfer knowledge between the different cancers. Of course, this is only transferring between cancers. If you want to transfer between cancer data and some other diseases like Alzheimer, that's a different story. So distribution of the data may be completely different than the transfer of knowledge. Even using meta-learning may not work well. I should point that out. So we did on different data sets also. So I will stop that. But so our main this grant is knowledge guided machine learning. So we want to, so with even meta learning can do better than transfer learning, we still want to be able to integrate knowledge into the model. So what we can do, we can transfer and integrate knowledges. So that's what we are looking at this different kind of ontologies. So the domain prior knowledge in biology is very rich. So for example, you have the disease ontology in the, so for the mimic data sets, you have all this classification of diseases into different hierarchy. And so this hierarchy, we can specify it as a graph and then enforce this relationship into the learning process. Since, for example, the Alzheimer and Parkinson's are closer. And so those, if we specify this kind of relationship, then your machine learning model can do better. So with that, we actually experimented with the mimic data sets, which is a recording of patients in ICU situation. And they have a lot of ICD-9 codes. So we represented this kind of relationship. They have this coding system into the diseases and in hierarchy based on the disease relationship. So we have a graph to design this relationship, and then we incorporate, we have this hierarchy graph to denote the relationship between different classes. And so with that, I will not go to detail of that, but I will just mention this method we published 2020 KDD paper is we designed this hierarchical graph in the disease hierarchy, and then incorporate that into when we train the embeddings, we incorporate that into the model. And so when we train the tasks, we can have the relationship, we can use that as a regularizer to enforce the relationship between tasks. So, and then we can perform better in prediction. So here is the results. When we look at the mimic, they have all different sample a huge number of classes and examples, but each individual, you have all these individual classes, how they allocate each. And then we compare the performance between all different meta-learning methods, and with our knowledge integrated that graph. And you can see here, when we integrate knowledge into the machine learning model, we do achieve better performance than all other meta-learning approaches. So that shows encouragement when using knowledge into the machine learning model. It does improve the performance. So I just hope to briefly introduce this and as an introduction, and experiments to show the knowledge-guided approach does improve performance. Of course, here in both, let me see what time. So in both scenarios, I use the cancer as a separate training and the mimic as a different training. So they are in different distributions. So, but a question, interesting question is, if I train on the cancer, can I, TCGA, can I use for mimic? I think that performance will not be very effective. I think that comes to heterogeneous distribution of data, how we can train models to deal with the heterogeneous situation. That's a more advanced research topic. So I just have a few minutes before the keynote speaker. So I just want to mention as a conclusion, I think there's still a long way to go, and there are many interesting problems in bioinformatics and health informatics. And I do think the close collaboration between the computer scientist and the domain expert is very crucial and important for effectively applying machine learning approaches in biology. So I will end my introduction there. And so maybe answer, a couple of questions before we introduce the session keynote speaker. Okay, so right here in the chat, there is a question: how does one account for variability coming from different data sources, technical noise, batch effects? So variability come from different data sources. That's a good question here. So you can see if it's the same distribution. So that question I can use this is if it's the same distribution. You can see here we have, when we test the meta-learning memo-based approach on TCGA, we experimented with the proteomic features. There are all, a lot of different features. So proteomics feature alone, this is the result. And RNA expression data, we got this result. Actually, RNA perform slightly better than proteomics here. And then we combined all clinical proteomic RNA, all the data here. This is the result we got. And you can see actually the performance is slightly lower. It's not obvious, but you can see from the ratio. So to answer that question, we look at the data, there are noises and some noises in the whole data sets. That's why the performance is a little bit lower. But you can see in the performance, it is actually pretty effective in incorporating all the noises and in the data set when we combine all the data. hope that shows that on here. Oh, we should move on time. So I think we at the end of the talk, if we have time, we can go back to the questions here. So I see if there's any obvious questions I can quickly answer. I don't been any effect to pan cancer to use. Have there been any effects to do pan cancer to use all in the cancer for questions that are independent for cancer type? So if I quickly answer that correct, so yes, so if we train the model using TCGA different cancers, so the purpose of using meta-learning is you train with existing data, you can quickly adapt to new data, especially rare diseases. The rare diseases, you have a few samples, then you can quickly adapt. I think if it's a rare disease in cancer, I think it should be effective if my understanding of the question correctly. What is getting classified? So the 3-way, 5-way means I give you 3 classes, and you should be able to predict it's one of the classes. 5 classes is one of the classes. So that's a typical few shots learning environment. In respect to interpretability, so with graph regularization, that's a good question. The interpretability, it was not a concern in that paper. I think we can do post hoc. If you are familiar with interpretability, post hoc interpretation we can do using this TADA network, but not a pre-hoc, interpretation in the model, because the integration and the deep learning is not it cannot be interpreted in the process, but we can do post hoc interpretation. Okay, so I think I quickly responded some questions after we have the 2 speakers before the break. I will come back to the questions. Let's first, I'd like to introduce our session keynote, Dr. Russell Schwartz as the session keynote speaker, and Dr. Russell is chair of the Department of Computational Biology in CMU and has a long time experience doing computational biology, and we are fortunate to have him to talk about his research today. And welcome, Russell. You can start.
Thank you, John. Thanks for having me here. Let me just share my screen here. Okay, I hope everyone is seeing my screen now. So I'm, as you said, I'm Russell Schwartz from Carnegie Mellon University. I want to tell you today about some of our work in cancer progression risks, so in particular, bringing machine learning to understanding some of the factors that influence the risk of progression of cancers, where progression here means the process of a cancer moving from different stages of disease development. For example, it might be from a healthy cell or healthy tissue to a precancerous lesion, precancerous to cancer, and then from primary cancer to a drug-resistant or metastatic form. And what we're really interested in here is what underlies the risk of progression. So what is, in a sense, the predictive information here? So it's perhaps a bit backwards from the way we might sometimes think of knowledge-guided machine learning. We're not using knowledge to try to create better predictors per se. We're using machine learning to try to understand what knowledge leads to better predictors. But to give some background into why we're doing this, I need to tell you a little bit about what the relevant knowledge is here. And that's something that comes about from largely the idea of cancer as a disease of evolution. So cancer, when I say it is a disease of evolution, what I mean is that cancer is governed by laws of evolutionary diversification or drift and selection. Typically, we believe a cancer would begin with a single cell that acquires some kind of mutation. Very often, maybe most of the time, that mutation will be something that itself induces a hypermutability phenotype. So that is, it primes the cell to more rapidly generate mutations, and that sets the tumor on the path, or really the healthy cell at that point on the path to ultimately becoming a tumor. It leads to expansion of heterogeneity as different cell lineages are rapidly acquiring mutations from one another, becoming distinguished from one another. And this genetic heterogeneity creates the opportunity for selection. So for some combinations of mutations leading to preferential benefits to the tumor and growing more rapidly, escaping controls on cell growth, escaping treatments, moving to new sites, and so forth. So this has been known for a very long time. I cite here Noel's work of 45 years ago, laying out these principles. And it's been an important way of thinking about cancers for many years. But I'd argue that where it really took a major leap forward was in the genomic era, when this understanding of cancer as a disease of evolution became a really crucial principle for being able to make sense of big data in the cancer field. And cancer is one of the hardest big data problems in the biomedical domain, I would argue, because it involves highly mutable cells. You have to keep track of an entire genome, and mutations anywhere in that genome might be relevant to tumor evolution. Different cells in a single tumor will have different combinations of mutations. Different tumors will have different mutations and different pathways through a selective landscape. And all of this needs to be interpreted using large, noisy, heterogeneous data sets, as well as in terms of very complicated biology that we don't fully understand. And evolution became a window through which we could make sense of a lot of this complexity and be able to extract some meaningful information from tumor genomic data sets that are so hard to interpret because of all of this complexity. And one of the key ways that happened was through the development of the field of tumor phylogenetics, which is basically just the idea of bringing phylogenetics or evolutionary tree inference to cancers. So being able to build evolutionary trees of tumors. Let me get a pointer here. So build evolutionary trees of tumors where we can take tumor cells, build a tree of those cells, identifying how they separate into different lineages, what mutations lead to this separation, and how that ultimately underlies the progression of a cancer. So all of that is something that's been studied for quite a while at this point, and there are now many variations on tumor phylogenetics. I want to briefly tell you a bit about this before I get to actually the interesting machine learning here. So my own group got into this field about 15 years ago now, working with actually pre-genomic kinds of technologies, in particular a technique called fluorescence in situ hybridization, or FISH, which basically just means fluorescently labeling pieces of DNA in a genome. And this turns out to be a useful thing to do in cancer studies, because one of the ways, maybe the main way cancers adapt selectively, is by gaining or losing copies of pieces of their genomes. FISH gives you a way of profiling this for a few pieces that you think are important, giving you markers that you can use to do phylogenetic studies, build evolutionary trees of cancer cells, and start to be able to tell these stories of how single tumors are evolving. Most of the field really took off, though, when it became possible to do whole genome sequences on cancers. So this is an example of a later method, really a set of methods that my group and many others have worked on, bringing these techniques initially to bulk genomic data, which essentially means genomic data on large populations of cells. That might be an entire tumor or a region of a tumor. This ends up creating a way of being able to see a lot more than you can see in those pre-genomic studies. In principle, see an unbiased view of all the mutation in an entire genome, but introducing another complexity that you have to deconvolve contributions from many different cells in each sequence analysis. This is typically posed as some sort of matrix factorization, but gives another way to identify a set of inferred cells in a tumor and how those might be related by a phylogeny that then gives you insight into what's functionally happening in tumor evolution. The field took another major leap forward with the work of Navin et al., who introduced single-cell sequencing to cancer studies. With single-cell sequencing, it became possible to get around the limitations of having to deconvolve data, which is a very noisy, low-resolution process, and became possible to look at tumor evolution at a single-cell level, looking at, in this case, these are profiles of copy number variation in a few different single cells, and get markers across an entire genome that could then be used to build yet other ways of constructing tumor phylogenies. Many of the most interesting methods today are multeomic methods. This is an example of yet another method that's combining single cell bulk sequence and FISH into a single inference by putting together a loss function that basically says that we want to infer cell populations, we want to infer how they're distributed over a tumor, we want to infer phylogenies among them, and we want to do this in a way that is consistent with how we think the biology works, as well as with all of our different data sources. And multiomics is a lot of where the interesting action is in cancer studies. And there's a lot you can do even without looking directly at the DNA. So you can build tumor phylogenies from RNA, for example. And this is yet another example of some work of ours. As with all of these things, we are far from the only group working on this, but work of an example of an RNA sequence phylogenies from our method, in which we are bringing in knowledge of how RNA gives us information about activity of different networks and pathways in a tumor. Just as an aside, that this could probably be a whole talk about knowledge-guided machine learning, the different ways we bring in knowledge about pathways we know to be important in cancers, gene modules and pathways we presume to exist, but don't assume we know about in advance, giving yet another way of building phylogenies and finding out how those are reflected in functional activity of tumor cells or populations of tumor and non-tumor cells within a cancer. And that is just to illustrate that there are many ways of doing this, each of which has complementary advantages for learning about phylogenies. Now, what I've told you about is just a few examples of phylogenies from my own lab's work, mostly because it's easier to talk about and not worry about asking for permission to use figures from my own work. But we are one small piece of a huge field of tumor phylogenetics that is active today. This, just to give you a taste of that, is a table I put together actually about a year ago now, listing just some of the space of DNA sequence-based phylogeny methods that are out there. There are many for different combinations of data types, different kinds of genetic variation we care about, single nucleotide changes, copy number changes in the genome, structural variations like translocations, rearrangements, inversions, and so on. And these are just a small fraction of the methods that were available a year ago when I put this together. It's an even smaller fraction of the methods available right now as I'm speaking to you. The important point is that there are a ton of methods out there for many combinations of data, many ways of looking at this, many ways of saying what it is we're trying to track in tumor evolution or how we propose to do that. And I say that by way of background because it's really leading to the main thing I wanted to talk about today, which has to do with what we learn from tumor phylogenies. What knowledge do they actually encode that is useful for us for making practical predictions about cancers? Now, when we, and I think most other people get into this field, we had maybe a naive optimism that what we would learn from tumor phylogenies is a window into the story of every particular tumor. We would be able to look at a cancer, we would be able to tell the story of how that cancer developed, what mutations were occurring when in the cancer, what evolutionary problems those were solving, suggesting how this overcame an evolutionary landscape to get to, let's say, tumor growth, to get to metastasis, and then be able to back out from there, how we could develop better diagnostics, better targets for therapeutics, and so forth, and treat maybe that particular patient, more likely patients like that one in the future. And I call this naive because that idea does not work out at all. One of the things you learn very quickly when you work in tumor phylogenies is that you learn absolutely nothing from looking at a single tumor phylogeny. And the reason for this relates to something I told you about back at the beginning of the talk. We believe in most cases cancers begin with the acquisition of some sort of hypermutability, very rapid acquisition of mutations. And as a result of this, most of what is happening in tumor evolution has nothing to do with the functional adaptation of the tumor. This is something well known from work of others, especially Christina Curtis's group in what's called the Big Bang model of tumor evolution. But essentially, tumor evolution is a highly stochastic process. Most of what is happening in evolution of one tumor is just random noise, and you can't tell by looking at one tumor phylogeny what is meaningful and what is random chance in that tumor evolution. Fortunately, though, that doesn't mean that there was no point in looking at this. When you start looking at large numbers of phylogenies, you do discover that there's actionable information in them. And that also is something that became apparent fairly early in these studies. This is just an example of a very early version of this from our work. This one was almost a decade ago at this point, using some of those fish phylogenies to try to ask, what can we predict about progression of a cancer? In this case, the particular question that we were trying to ask is, can we look at primary cervical cancers, so the initial tumor that a doctor sees in a clinic, and identify the cancers that will progress to metastasis? And the short answer was that if we take information from the raw data that goes into building these phylogenies, basically different ways of slicing up the raw count data that we use to build the phylogenies, we do no better than chance in producing predictors of whether we're looking at a metastatic or a non-metastatic cancer. But if we look at this through the window of tumor evolution, so we build our phylogenies, we use those to extract features that we can derive from the phylogenies in different ways. So things that tell us about propensities to mutate by particular kinds of mutations, propensities for overall mutability in the tumor, propensities to evolve in, let's say, ways that promote more genetic heterogeneity or less. There's a lot of information in that lets us make accurate predictors of which tumors go on to metastasize and which don't. And I'm not going to go into many years of work that followed up on that, but it basically showed that as we build better and better models of tumor evolution, these predictors become more and more powerful. Essentially, there's information in the evolution of these tumors that is ultimately encoded in the raw data, but you have to bring in the knowledge that you're working in an evolutionary system, the expert knowledge of how to extract information about the evolutionary process to make use of that. And that was a very general point that came up again and again in our studies in this field and others over the years. And that brings me to a summary slide I put together here that's trying to summarize a very large body of work over many years, but just to get to the point that's really underlying the main thing I wanted to talk to you about today. And that is summarized as follows. There are lots of ways of building tumor phylogenies, and quite broadly across these ways of building tumor phylogenies, we observe variation in the evolutionary processes of these tumors. Different tumors have different propensities to evolve in different ways. There are different hypermutability phenotypes. Some tumors have higher mutation rates than others. Some have higher rates of point mutations. Some have higher rates of copy number mutations. There are different subclasses in each of those. Some have higher rates of one kind of point mutation versus another, one kind of structural variation versus another. And this is something that shows up every way we look at tumor evolution, every kind of cancer we look at. Not only that, but that variation in mutational phenotype, so how a tumor is evolving, is quite broadly predictive of progression outcomes in cancers. Again, in our work and the work of many others, there are many ways we've asked this question on different cancers, different kinds of progression phenotypes, different phylogenies, different data sources, and it comes across quite broadly that how a tumor is evolving gives you predictive power for how it's going to progress in the future. And that's really the key takeaway of everything I've talked about so far, which is leading into the main point I actually wanted to get to, and the main thing I wanted to discuss with you over the remainder of this talk, which is the real question we want to ask here, which is how important is that knowledge? In terms of evolutionary theory, evolution has two pieces. It has drift, or the generation of genetic diversity, and it has selection. So picking diversity that gives an advantage of growth, or in some other way, an advantage to the thing we're looking at. In this case, the thing we're looking at being tumor cells. And in most studies of phylogenetics, most studies of evolution, indeed most studies of cancer evolution, the focus is largely on selection. We're largely looking in the cancer sense at driver genes, so genes that are believed to have some sort of functional advantage for the cancer. And in most studies of phylogenetics, drift is generally assumed to be a generic background process. It's something we're usually assuming is not a variable that we're looking at in our process. It's something that is just sort of happening in the background and is giving us a window into selection. There's certainly exceptions to that in both species and tumor evolution, but by and large, that is how we look at these things. And I would argue, based on the work we had seen so far, that in cancers, variability and drift is a very important piece of the puzzle of understanding cancers, and cancers give us a window into how that is occurring. So we want to look at this question of specifically how important is drift in cancers? How important is the background process for generating mutability versus the specific spectrum of mutations that occur in any particular cancer? And when I say how important, what I mean is how much do they control the risk of cancers moving to later progression stages? And I'd argue that that's an important question for a couple of reasons. One of the reasons that's an important question is, of course, that we study these things because we want to help patients. We want to save or improve patient lives. And I would argue that it's quite important if drift is a source of a large fraction of the risk, because we believe that variabilities in the drift process are a very early step in tumor evolution. So we would believe that in most cases, the acquisition of a hypermutability phenotype is something that is occurring years before a cancer is visible, really years before it even is a cancer. And that most likely, when you see a tumor, you're looking at something that has spent several years, maybe many years, silently acquiring high rates of mutability before there's any visible phenotype of tumor growth. So if drift is important, it's suggesting that there's actionable information very early in tumor evolution, even before there is a cancer present. And we'd argue that that's something that we would want to know about. I'd argue this is also an important question for basic research into well, not just cancers, but into evolution. So evolution has been a crucial guiding principle for understanding cancers, but I think it's less appreciated outside the cancer genomics field that cancers are also a great system for understanding evolution and can teach us some things that are very hard to learn in other contexts in evolutionary theory. Cancers are an unusual model system in evolution in that we get to observe millions of examples of navigating essentially the same selective space multiple times. And we can start to see patterns of how that's done in different situations and understand something I think actually relates to what one of our later speakers, Bin He, is going to talk about, how reproducible is evolution. Cancers are a great system for asking that question. And this is an example of one of the unusual things about cancers where we have this variability in drift processes that's hard to observe in other natural systems. So all that is really setting up the question we wanted to ask here. And that's where this really becomes a talk more about machine learning, that we want to use machine learning as the window of asking this question: how important is the evolutionary process and variability in that process and underlying cancer risk? Our overall paradigm for asking this question is to build evolutionary trees of cancers from DNA sequence data and use these to extract sets of features that describe the evolutionary process. Some of these are features that are describing things like overall rates of mutability, overall rates of particular subkinds of mutability, as well as overall features such as tree shape statistics that can describe whether we're looking at a process that leads to a lot of heterogeneity or low amount and so forth. And then we're trying to ask, how important are these features? How much of our ability to predict comes from this kind of knowledge specifically? And we ask that in contrast to two other sources of knowledge. We have driver features. So driver features are features that tell us about mutations in particular genes that are important to cancer. So specific mutations. In the evolutionary features, we're abstracting away what particular genes are being hit. We're looking at overall processes. In the drivers, we're paying attention to the genes. Now, we would expect drivers to encode some of the information in the evolutionary features as well, because ultimately hypermutability phenotypes come about because of mutations in driver genes. So in principle, those variabilities in evolution are redundantly encoded in the driver genes, but there's also information about selection that's lacking from there. We also look at clinical data, so basically metadata on things like patient demographics, things a doctor could observe in a clinic, things a pathologist would observe examining a tumor sample or biopsy in their laboratory. And then we want to ask collectively, out of all this information, how much of the risk that we can predict comes from the evolutionary information. We ask this in a number of different variations using two data sources: the Cancer Genome Atlas that we heard about in the previous talk, and the International Cancer Genome Consortium, a large international consortium, both of which are providing large amounts of tumor data. And we asked this question in a few different variations. We looked at two kinds of sequence data, whole exome and whole genome. Whole genome is much better for asking this kind of question because we can't see a lot of kinds of mutability in whole exomes. A whole exome means just only looking at the protein coding regions effectively. But we have to look at whole exome as well because the cohorts for whole genome sequence are very small. We simply don't have enough samples with whole genome sequence. We wanted to look at these bigger whole exome cohorts. We looked at two cancers in this study, breast and lung cancer. And we looked at two outcomes: overall survival, which means how long does a person survive after they've been diagnosed with the cancer, and disease-free survival, which is how long until they either pass away or have a recurrence of their tumor. And that gave us a number of different variations for asking this question. Now, I don't have a lot of time, so I'm not going to go into great detail about the actual machine learning methods we used. Basically, we used a kind of heuristic feature selection based on stepwise forward discriminant analysis, combined with a specialized kind of machine learning for this sort of survival data known as Cox regression with L0 regularization. And if we want to use this, we basically have to show first that we can make predictions. This is just showing that we can indeed, using all of those features, make accurate predictions. This is in particular showing that those models give us a way of accurately stratifying patients into good and bad prognosis subgroups and getting a significant separation. We do a lot better with the less informative but large cohort whole exome sequence than the more informative but small cohort whole genome sequence. Some of those are kind of at the threshold of significance here, but we are at least able to build good classifiers. And that lets us start getting to the question of what we actually learn from these classifiers about the importance of particular feature types. We started by just asking for individual features, how important are they? And this is a way of asking this in terms of over many random replicates, how often does a feature show up as significant following the feature selection process? And the short answer is that features from all three of these classes show up as significant. This is the breast cancer whole exome sequence. We get evolutionary, driver, and clinical features showing up. In general, the clinical features are the best, which is what we would expect. fact, clinicians have done a good job of finding a feature set, but there are predictive features from all of these classes. And we see similar things when we look at the whole genome sequence, similar things when we look at the lung cancer, which I'm not showing here. Individual features actually drop in reproducibility from the evolutionary class when we go to whole genome sequence, which I think is a combination of more redundancy among the features when we have more of these features available, as well as the smaller cohort sizes, making it hard to get significance on ones that are kind of at the threshold there. So that's telling us that there is information in all of these feature classes. In particular, related to our question, there is information in the evolutionary features. But our question is hard to answer nonetheless, because there's a lot of redundancy among this information. And that's captured in this matrix of cross correlations, basically pairwise correlations between different features. This is showing in red highly correlated features. So the two axes of these matrices are just the feature sets aligned against one another. So the ones that pass the feature selection. And essentially what we see is a partition into a few submatrices. The clinical features pretty much partition into one submatrix with relatively low correlation internally and with the other feature sets, which is just suggesting that clinical features work well. Clinicians and clinical researchers have done a good job of finding a small number of things you want to check that give you a lot of relatively non-redundant information. We next see a couple of submatrices that turn out to correspond to the evolutionary features, and they pretty much partition into one group for the structural and copy number variation features, another for the point mutation features. There is substructure within each of these. It's harder to appreciate on this diagram, but to a first approximation, there are two axes of variability corresponding to those two kinds of mutability. And that's not so surprising in light of the prior literature. Driver features show up as their own submatrix, and we have a kind of weak mutual correlation among all of these submatrices. And we think that's largely occurring because of a kind of proxy effect, where if you have a highly mutable tumor, you will see a lot of driver mutations. And if you look at a tumor with a lot of driver mutations, more likely than not, you're seeing a highly mutable tumor. So that's why we think we're kind of getting largely this background pattern of mutability. And we see similar things in all of the other cases. And there is evidence for a lot more correlation structure here that we're not fully exploiting. I just show you an example of this by doing a dimensionality reduction, taking those evolutionary features into a two-dimensional space with t-SNE. And essentially what this is showing is how point clouds of the tumors correlate to the progression outcomes here. It at least looks like superficially there is a good correlation there. In the breast cancers, maybe islands of good outcomes in spaces of largely bad outcomes. In the lung cancers, it almost looks like the opposite, islands of bad outcomes within spaces of good outcomes. But at least to a first approximation, it does appear that probably we're seeing these same two axes of features here, more or less point mutability and structural variation mutability. And there's a lot of information just in knowing where a tumor sits on those two axes. And that's not so surprising. It's been known for a long time. More mutable tumors have worse outcomes. Tumors that are mutable by what's called chromosome instability, so basically a propensity to have structural variations, have worse outcomes. And that ultimately lets us come to the real question we're trying to answer here, though, which is, among all those features, how important are the evolutionary ones? So we decided to quantify this in terms of hazard ratios. Essentially, how much information does a subclass of features give us to figure out someone's risk of developing a particular progression outcome in this kind of time series survival data. And essentially what we're able to find is that every class of features has significant predictive power. It depends exactly how you ask the question, how much predictive power there is, but evolutionary features have power, driver features have power. When you put them together to get what we call genomic features, which is just shoving the evolutionary and driver features together, you have more power. So each of those has predictive power, but each has some non-redundant predictive power, which is suggesting that we get something by looking at the mutations through the window of evolution. The clinical features are consistently the most powerful, but when we add genomic data on top of that, we get an extra boost of power beyond that. So again, there is more more to the tumor than we can see from the clinical data. And we can use that to visualize an answer to our question, a kind of rough quantification of how important these features are, based on how much of the progression risk is contributed just by the evolutionary features. These circle plots are showing in orange the amount of hazard coming from the evolutionary features, in yellow the amount of extra hazard we get when we add the driver features beyond those. So effectively taking the drift in the orange features and adding information about selection on top of that. And then the red is bringing in all the kind of extrinsic factors beyond the genome that would be available to a clinician, things like patient age, demographics, pathology reports, and so on, which contains quite a lot of predictive power. And that lets us draw a very rough approximation of an answer here, that more or less the amount of importance we attach to the drift is the amount of orange in each of these circles. And very roughly, we'd say it's about 1/3. And let me emphasize here that I would not attach any importance to whether it's 1/3 or 1/4 or 2/3 or whatever. The important question here is really, is it roughly 0? Is it pretty close to 0? Is it pretty close to 1? Or is it something in between? And I would argue that this study is saying very clearly it's something in between. Drift is an important part of underlying progression risk. That is variability in drift processes, variability in somatic mutation mechanisms. It's not everything, but it's far from nothing. And again, I would argue that is a conclusion with important implications for cancer research and treatment, because it's suggesting that there may be actionable information long before we actually have a tumor readily apparent, if we can figure out how to get at that information reliably. I know time is short here. I think I really need to wrap up. So let me just say that one of the reasons I like to talk about this is because I think this study could be done so much better than we did it. There was a lot more to be done with bringing this to other phenotypes, other data types, other kinds of cancers with actionable progression questions. There's a lot more that could be done by bringing more sophisticated evolutionary theory to this problem, and a lot more that could be done by bringing more sophisticated machine learning, and especially better uses of knowledge in machine learning to this problem, including knowledge of networks and pathways that we know are important in these cancers, ways of designing machine learning models simply to use the knowledge that we know these cancers act through biological systems, that we know they act through tumor heterogeneity and evolution, and other ways of taking advantage of the structure of the data space, for example, through more sophisticated manifold learning ideas. So let me just close there by acknowledging the contributors to this work, especially my student, Yifeng Tao, who's lead author on a lot of what I described to you today. Yifeng is actually defending this afternoon, so I hope you enjoyed his work. We're really proud of him. Thanks to others in my group who contributed, and to work from my collaborators' labs, Lab of Jen Ma at CMU and Xinghua Liu at the University of Pittsburgh. Thanks to our funders, and thanks to all of you for your attention.
Thank you, Russell, for the great talk. Let's see the questions. Okay, I'm going to read the first question. Russell, have there been any comparative effectiveness studies comparing different tumor phylogenetic methods against one another given the same data?
Yes, that's something that comes up really most of the time when someone designs a new tumor phylogeny method. It's just expected that you'll benchmark it against other methods. It can be a challenge because different methods are constantly being designed that use a new kind of data, for example, and then there's nothing to directly compare it against. But that's something that we would routinely do when we develop a new method. We compare it as best we can to other methods and anyone else in the field doing that. I'm not sure if I could cite a particular review study where someone has just done that comparison more broadly. I'm sure they exist, but off the top of my head, I can't think of one.
Okay, thanks, Russell. The second question is, Russell, for the explained variation of progression risk, the genomic features appeared to be tested first. and then the clinical features. If the order were reversed, question mark.
Yeah, so you could reverse the order that we're doing that analysis, and it would really be asking a different question. Our concern here really was how much information do we get just from the evolutionary features, but then we would be getting that question, then the question of how much information do we get just from the genomic features. If we had done the clinical first, those have redundant information with the genomic features because they do include some assays for important genomic effects as well as other information. So it would really be asking a different question about how much we get just from the clinical data and how much does genomic data add on top of that. And you can kind of get that conclusion from the things I presented, but you would end up with a somewhat different answer that really the clinical data contains most of the information, but then genomics is giving you a boost beyond that. So it does allow you to improve your accuracy over current clinical metadata.
Okay, thanks, Russell. We're just, at the time, 11:30 for the next speaker. So thank you, Russell, for your effort and contribution to the session. And great talk. And great to see you.
Likewise.
Okay. So I'd like to introduce our next speaker, Dr. Tumor Kavesi, right? I hope I pronounced your last name correct, Kavesi, from University of Florida. And Tumor is associate chair of the Department of Computer Science there. And Tumor has a long list of contribution to the bioinformatics community. I will not list there, so please read his bio list posted in the website. And we are fortunate to have Tumor here to give a talk. And Tumor, so it's all yours.
Thank you. Thank you so much for the kind introduction. Share my screen. Is my screen visible to everybody? I don't?
It is.
Okay, perfect. Right. Thank you and welcome to my talk. I'm going to be talking about the motif counting problem, which is a very fundamental problem in understanding how biological systems work. And particularly, I'm going to be focusing on how the evolution of biological networks influence the motif counting problem. I cannot see my own slides. Okay, as we all know, biological networks describe how different molecules interact with each other. And depending on the type of the molecules that interact and the kind of interactions that takes place between them, we categorize biological networks in several categories, such as metabolic networks or protein interaction networks. But usually we represent these biological networks, as you all know, using graphs. And the nodes in the graphs are typically going to be representing the molecules, and the interactions between them are going to be represented by edges between the nodes. Now, under this mathematical model, a motif is simply a small graph pattern such that this graph pattern appears in a given target network much more frequently than you would have expected in a randomly generated network of the same size. Of course, in order to talk about whether a motif appears so many times in a given network, the first thing that we need to mathematically define is how do we count a motif in a given network. In the literature, there are typically three different ways to count motifs. I'm going to call them F1, F2, and F3 measures. And I'm going to explain them very briefly on this toy example. So in my example here on the top, what you see is a motif pattern. And at the bottom is a small hypothetical network. Now let's start with the F1 measure. The F1 measure simply says that, hey, I want to find the I'm only interested in the number of possible ways to place this motif on top of the target network. For example, I can place my motif on the network like this, or like that, or like that. So there are 3 different ways to do this placement. So the F1 measure says that my count is equal to 3. Now let's take this motif and make it slightly bigger by adding a new node and a new edge to the motif. Now, how many times does this motif appear in the target network? You can notice that there is only 1 copy over here. So the F1 measure goes down from 3 to 1. Now, at this point, you're possibly thinking that it's not surprising that the motif count went down because my motif actually got larger. So larger motif means more constraints, so the motif count is going to go down. But actually, that's not the reason why the motif count went down. To understand this better, let's just do the opposite and make our motif slightly smaller, this time by reducing a node and an edge. Now, let's see how many copies of this motif I can place on the target network. There's 1 copy here and another copy here. So my F1 measure is going to be equal to 2. And from this simple example, we observe that as we grow the size of the motif, the number of times that we can observe it according to the F1 measure sometimes goes up, sometimes goes down. So it's not monotonic. When this happens, we say that this measure does not have the downward closure property. And when the downward closure property is violated, it's really bad for a lot of problems, a lot of questions that you can ask on the biological networks, including all the prediction and the qualification, classification problems on biological systems. F2 and F3 measures deal with this problem by adding some constraint on the measure. So F2, for example, F2 measure says that I'm going to place my motif on the target network with the constraint that these placements do not share any common edges. So edges have to be independent of each other. So with this constraint, I can place a motif here and another copy over here. So 2 copies in total, and they do not have any common edges. And this is the largest number of placements that we can have on the target network. So my F2 measure is equal to 2. The F3 measure is more stringent. It says that not only the placements of the motifs should be independent in terms of the edges, but they should also be independent in terms of the nodes. So I'm not going to allow any overlaps between the nodes and edges. When you have this constraint, there's only 1 copy of the motif that I can place on the target network, and any other copy that you can place is going to be overlapping with this. So the F3 measure is going to be equal to 1. So when we put all these 3 measures together, we observe that the first one does not have the downward closure property, but F2 and F3 do have the downward closure properties. We can prove that mathematically. So I'm going to be focusing on the F2 and F3 measures in the rest of my talk. And particularly, I'm going to be focusing on the second one because there's like algorithmically very, very tiny difference between the F2 and F3 measures. But regardless of the measure that you pick over here, all these 3 motif counting techniques are NP complete, computationally very expensive. Now, those of you who do theory among you are going to hate me for saying this, but I'm going to say that F2 and F3 measures are going to be are harder problems than the F1 measure because you have to solve not 1 NP complete problem, but 2 NP complete problems for each one of them. But at the end of the day, it's still NP complete. Now let's talk about a little bit about the evolution of the networks. The network topologies are not static entities. The network topologies change over a period of time. And these topological changes take place for many, many reasons, like stress conditions, like extreme heat, cold, oxidative stress, lactose stress, aging, or many other reasons. But regardless of the what the reasons behind the evolution is, the network topology changes over a period of time. We mathematically represent this evolution of networks over a period of time as a sequence of events where each event is an edge insertion or an edge deletion to the network. Now, we can also consider this as node insertion, node deletion as well, but it's essentially the same thing when you take all the nodes in all the instances that you observe together, take their union, and whenever that node is not observed in a certain state, it just becomes an isolated node with no edges. So I'm going to represent it as a sequence of edge insertions and deletions. And mathematically, I'm going to represent it using a tuple. The first entry in this tuple, G0, is the topology of the initial network with all the nodes and edges. The second entry is a sequence of edges where each edge represents the edge that gets inserted or deleted from the previous state. So if you have that edge in the previous state, then you remove it. If you don't have it, then you insert it. So this way I can represent a sequence of n dynamic network states using this tuple. Under this definition, I redefine the motif counting problem as follows. So given a motif topology and such an evolving network with n different network states, my problem now is to find the number of times this motif is observed in each of these instances of the evolving network. So if there are 1,000 states, I want to find the motif count in each and every one of those 1,000 states of the network. Now, I just said in the previous slide that this is a NP-complete problem. It's computationally intensive for just one network, which means that I definitely do not want to compute this over and over again for thousands and thousands of network topologies. And what I'm going to describe is a strategy to do this incrementally by just using the current motif count plus the observed changes from one state to the next state of the network. Just a second. All right. So to explain this concept, I need to first explain how we calculate the motif count, the F2 measure, in a static network first, which is the initial state of the network. I'm going to explain our algorithm, which is an algorithm that works in 2 steps. I'm going to explain it on this small example. So here we have a small hypothetical motif, and here we have a target network with 6 nodes and 10 edges. In the first step of our algorithm, what we do first is we find all possible placements of the motif on the target network, regardless of their position, regardless of whether they overlap or not. I ignore all the constraints and I find them. For example, one possible placement is edges 1, 2, and 3. So I find this and I keep that information. And as I do this, I start creating a new graph, and I'm going to call this an overlap graph. In the overlap graph, each node corresponds to one possible placement of the motif on the target network. So for example, this one corresponds to H1, that is edge 1, 2, and 3. So I repeat this. I find the next placement of the motif, which is 3, 4, 5. So I create a node for that. And then I insert an edge between these 2 nodes, meaning that these 2 placements of the motifs have a common edge. They are overlapping with each other. In this case, it's the 3rd edge that is common to both of the embeddings. So just like that, I continue finding new embeddings and grow my overlap graph with new nodes and new edges. And this is what I have at the end. Next, I need to select a subset of these placements of the motifs such that everything that I selected is independent of each other. They do not have common edges, plus it's as large as possible. So to do that, I make the following observation. I say that, hey, if I select any node, any placement from here, I cannot select any other node that has an edge with that node. So every edge for a given particular node is a constraint which prevents me picking from something else. So from this observation, I make a greedy decision to pick the node with the smallest degree, which is either H1 or H5 in this example. So I randomly pick one of them and put it in the result set. Now notice that H1 is connected to H2, which means that I cannot select H2 anymore. So I remove this from the overlap graph and I repeat the same process. Now my options are 3 and 5. So I pick one of them, put it in the result set. 3 is connected to 4, so get rid of 4. Now I have only one node left, so include it in the result set. And there you go, we have our solution, which is 1, 3, and 5. I can place my motif in 3 different places without any overlap. So that is the algorithm for the static network. Now, how do we extend this for dynamic networks? To do that, I'm going to need an auxiliary data set. In this auxiliary data structure, what I have is a list for each edge. In this list, for each edge, I maintain the list of embeddings which contain that particular edge. For example, let's focus on the third edge. The third edge is contained in the first placement and the second placement, H1 and H2. So the list contains H1 and H2 over here. Now let's start dealing with the evolution of the network. First, I'm going to look at what happens when we remove an edge from the target network. So let's remove an edge. In this case, it's going to be edge number 7. Now, when we remove an edge, what happens is that any motif instance in which this edge that I have removed participates is going to disappear from my solution set, which is 3 and 4. So these two are gone. Now remember that my optimal solution at this previous state was 1, 3, and 5. Just keep that in mind. So I remove 3 and 4. I go and update my auxiliary data structure, remove 3 and 4 wherever they appear. And then, because 3 is a part of the solution, I need to reduce my motif count by 1. But maybe I don't need to reduce the motif count by 1 because H3, when it was in the solution set, it was adding a constraint that I cannot add any of its neighbors. So maybe one of its neighbors, which could not be in the result set before, now can be in the result set. So to find it out, I look at this. It says that if there's a solution that can be included in the result, then it should contain 4 or 6. So I look at 6. Now 6 is empty, so I cannot include anything from here. Then I look at 4. 2 is available. So maybe I can include 2, but can I include it? Now H2 doesn't conflict with 3 anymore, but it still conflicts with 1. So I cannot include H2 anymore, H2. So my motif count goes down by 1. Now let's see what happens when we insert an edge. So I'm going to insert an edge and call it edge number 11 over here. Now, when I insert an edge, what can happen is that I may produce a new instance of the motif. Of course, to find out if we generated a new instance of the motif, we don't want to solve the motif counting problem from scratch again. That's too expensive. I want to do it quickly. So to do that quickly, I first look at my motif topology. What is the diameter of this motif? The diameter is the length between the farthest away pair of nodes. So the distance between these two nodes is 1, these two nodes is 1, 1. So the largest one is 1. So it means that my diameter is equal to 1. So what does it mean? It means that if there's a new solution, it has to be within a distance of 1 from the newly inserted node. So if there's a new solution, it has to be a combination of the red and the green nodes. So I only restrict myself to these colorful edges, and I observe that there is actually one new instance of the motif, which is 5, 9, and 11. So I'm going to call it H6. I generate it, I insert it in the overlap graph. I update my auxiliary data structure with the new list. Now 6 appears in, also contains edges 5 and 9. So I need to update those two as well. And then I have to check, hey, is this new motif placement, is it possible to include it in the result set? So remember that my solution was 1 and 5. So I need to check if this new embedding is overlapping with any one of them. So there's an edge here. So it means that unfortunately I cannot include that. So my motif count doesn't change. So this is the algorithm in a nutshell. So next I'm going to show you the evaluation of this algorithm. So we evaluated our algorithm on synthetic and real data sets. For synthetic data set, we generated random networks using 3 different random network models. Erdős-Rényi, Watt-Strogatz, and Barabasi-Albert models. We generated networks from 1,000 to 5,000 nodes with average degrees from 10 to 20. For each one of these combinations of parameters, we generated 10 random networks. And for each of those random networks, we created a sequence of 1,000 evolutionary steps by just doing random shuffling of edges using the degree preserved shuffling like this. For the real data set, we use a data set that we generated in one of our previous papers. So this is a data set from human embryonic stem cell and its evolution into different tissues. And we particularly focus on pancreas and liver. So there are two pathways of evolution from stem cell to pancreas, stem cell to liver in our data set. The size of this data set is roughly like 2,000 nodes, 14,000 edges. And one of the pathways contains about more than 3,000 network edit operations, edge insertion and deletions. And the other pathway contains more than 2,000 edge insertion and removals. These are the motifs that we tested our algorithm on. So we use these motifs because in another paper before we have proved that this is the minimal subset of motifs which can be used to produce any other motif of any topology of larger size. So there is no other subset which is smaller than this, whose combination can produce every possible topology. So in this experiment, we measured the running time. What you see as the red line, horizontal line over here, is the running time of calculating the motif count only for the initial state of the network. So if it was a static network with only the initial state of the network, this is the running time. The blue horizontal line shows you the total time to calculate the motif count only for two states of the network, which is the initial state and the final state of the network after 1,000 edit operations. What you see in between, this diagonal line, is the running time of our algorithm for calculating the motif count at each and every state of the network for 1,000 consecutive network edit operations. So what we see here is that in all these three random network models, Erdős-Rényi, Watt-Strogatz, and Barabasi-Albert models, we see that at the beginning, we pay more cost than calculating the motif count just once, because we need to maintain all this additional data structure, auxiliary data structures. But then once we pay that little cost, we can incrementally update the motif count with such a negligible cost that we can do this operation 1,000 times, and still it's not going to be as much as doing it only twice. So the cost here you see at Barabasi-Albert is slightly more, actually relative to the red and blue line, it's more than the Erdős-Rényi Watt-Strogatz methods. And this is happening because in the Barabasi-Albert model, the nodes are going to be well connected to each other. There's going to be some hubs. As a result of that, there's going to be a lot more instances of motifs in the target network of the same size under this model than the other two models. But nevertheless, we can calculate the motif count for 1,000 steps and still spend less time than calculating it only twice. Next, I'm going to show you the accuracy of our method at the most disadvantageous stage of our method, which is the final stage. So we make 1,000 decisions, and some of them are heuristic decisions. How bad is our result after 1,000? The network has evolved at 1,000 steps. How much does it deviate from the optimal result, which calculates it from scratch at the final stage? And we report the results for different number of nodes, different network topologies, and 4 different motifs that I showed you before. In all of them, what we observe here is that our method has an accuracy of, this is the lowest point, 96%, up to about 100%, so very high accuracies. And we also observe that for larger networks, our accuracy seems to be doing better. This is the accuracy results for different network densities, degree from 10, 15, and 20, the average number of edges per node. Again, the accuracies are very high. So it's about 96% at the lowest point and about 100% at the highest point. It ranges between the two. And with increased network density, our accuracy seems to be getting better, especially for the Barabasi, Albert, and Watts-Strogatz methods. Next is the real data set. These are the two pathways, stem cell to pancreas, stem cell to liver evolution. Now, I want you to notice that the number of dynamic steps here is much more than the number of dynamic steps that we had for the synthetic experiments. It's more than 3,000 here, more than 2,000 over here. And you're going to see that our results for the real data set are actually better than our results for the synthetically generated data for any of the three random network models. So although we calculate the motif for so many different steps, we are still below the total cost for just two static network motif counting operations. This is our accuracy for the two pathways for the four different motifs on the real data. Again, the results that we are observing are better than what we were observing for the synthetic data. Our accuracy at best is somewhere between 97, 98% accuracy and around 99% accuracy. So in conclusion, we show that the motif counting problem, which is a very difficult problem, NP-complete problem to do only for even a static network, it can be solved incrementally for evolving networks with very, very negligible cost and very high accuracy. Another observation that we have is that the bias degree distributions, especially those that we observe in real networks, are actually benefiting the algorithm that we have. So we do better for those kind of networks than the synthetically generated networks. So these are my collaborators, my students, Kingshuk, Mahmoudou, Christina is a faculty in our department. And I'd like to thank you all. You can take questions.
Thanks, Timer, for the great talk. We are at the time of break, but there are two short questions. The first one, the F2 and F3 measures of motif counting are mathematically advantageous, but are there advantages for biological interpretation?
Mm-hmm. That's a very good question. So what we showed in different papers, several papers, is that the independent motif embeddings are going to show you the possible functions, the possible number of times that the ability of the system to function. So what I mean by that is the following. I guess I need to go back to the motif topology. Okay, let's go back to this example that we have over here. Okay, so what I mean is the following. Assume that this is a system with a limited resource. So these are gene products, and you have a limited product. So when the motifs are representing the functional units, for example, feed forward loops or feed backward loops, so whatever the underlying function is. So the purpose of making them edge independent or node independent is that we consider these as limited resources. So once you consume a certain resource, the next one should be independent of that, because those products have been functioning in the process through a different combination of the nodes. So the more copies of that shape that you have, the more of that topological function that you can serve in the underlying system. So it gives you the set of the tasks that can be carried out independently in the underlying biological system. While the other one shows you not the independent one, the F1 measure shows you not the independent one, but the number of alternative ways to do it at different times. So I'm not saying that F1 measure or F2 or F3 measure is biologically, one of them is bad, the other one is good. They all have their meanings, but they have their own meanings. They mean different things. The way that I perceive the F2 and F3 measures is to understand how the functions are performed with limited resources. F1 measure, I understand it as how the functions are performed with unlimited resources. I don't know if that answer was clear.
Yeah. Tamer, so thanks. The second, maybe very short. How does this approach extend to graphs with directional edge?
It is the same algorithm. I mean, essentially, if you go over so there's really no difference between the directed and undirected ones. The only reason that I'm sticking to the directed ones is because of the kind of networks that we tested on, plus the motifs. So none of these steps in the algorithm change for directed or undirected networks. So we use undirected networks in this work only for practical reasons.
Okay.
I cannot think of anything that will change between directed and undirected.
Okay. Tamer, thanks. Thanks again for your talk. And we ran over 5 minutes. So, so this ends the first part of the session for 2 speakers, and we have a 10 minutes break. So we will resume. Well, Eastern time is 12:10, Central time 11:10. So in 10 minutes, we will resume, and we will switch moderator to Dr. Kevin James to introduce the other 3 speakers. Okay, and see you in 10 minutes. So thank you, Tamer and Russell, and great to see you.
It was my pleasure.
Yeah. See you in 10 minutes. Bye.
All right, welcome back, everybody. My name is Kevin James. I'm a professor in the Department of Biomedical Engineering here at the University of Virginia, and it is my pleasure to host the second half of this session for knowledge-guided machine learning and translational biology. And I'm happy to introduce our first speaker, Dr. Jisoo Park, computational biologist in oncology at the Genomics Institute of Novartis. So Jisoo is our first industry speaker at this year's workshop. Thank you very much to her employer for allowing her to come and speak. And before Jisoo begins, I'm going to remind that we're doing question and answer through Slido. So please connect on the link. If you have questions, you can ask them of Jisoo in real time, and then we'll address them at the end of her talk. And so Jisoo, floor is yours. You want to begin sharing your screen?
All right, thanks for the introduction. Do you see my slide without a problem?
Looks terrific.
All right, then I'll take it away. Thank you. Hello, everyone. My name is Jisoo Park. And as introduced, I'm a senior computational biologist at Novartis. And I'm going to introduce my postdoctoral study that I published last year with Trey Decker at UCSD with the title of Decoding Cancer Cell Maps to Guide Precision Medicine. This work started to tackle the problem that only a small portion of the drugs that go into the clinical trials is eventually approved by FDA. The low success rate of the clinical trials is often due to the incomplete understanding of the mechanisms governing the drug response or improving the cell sensitivity to drugs. To solve this problem, we decided to utilize the deep learning models with the superb predictive power, as many researchers have been making a great deal of interest in applying advances in artificial intelligence to tackle various problems in biomedicine. For example, like the deep learning models in radiology. In 2019, deep learning models started outperforming human radiologists in diagnosing various lung diseases based on X-ray images. And more importantly, the models also accurately localized the diseases in the images. The ability of the deep learning model for localizing the disease in the images is very important because it explains why the model made such a prediction, and that information is crucial for doctors to decide the treatment options. And this is exactly what we try to achieve in the context of cancer genomics, to predict the drug response and explain the mechanisms underlying it in terms of the cell mutations. However, the problem gets more complicated when we enter the world of cancer genomics because the patient tumors are very, very heterogeneous, as shown in the scatter plot. It shows a portion of chromosome 17. The scatter plot shows a portion of chromosome 17, which is on x-axis, and then you can see the patients on y-axis has mutations in different genes except TP53. TP53 is shared by so many patients, and the fact makes TP53 a bad pattern to stratify the patient tumors. In addition, there are more complicated problems that the driver mutations for each tumor are known to be different, and cancer pathogenesis is a very complex process where various mechanisms work in a combinatorial way. For these reasons, it's almost infeasible to identify shared patterns among different cancer populations or cancer subtype populations using individual mutations, and therefore performing such an analysis at the pathway level should be the way to go. So to implement the whole idea, we came up with the deep learning-based framework, which enables the analysis of cell mutations at the pathway level. We call this framework DrugCell, and I will introduce it in the following order. I will first present the overview of the DrugCell's framework and show you the predictive power of DrugCell. Then I will present our proof of concept analysis using paclitaxel and how we interpreted the model's prediction. And then I will talk about two studies that validate our findings via DrugCell. First of all, this is the overview of the model structure. Unlike conventional deep learning models, drug cell model has two branches. One branch on the left takes genotype of the cancer cell lines defined by mutations, and the other on the right takes chemical structure of drugs as an input. Two branches are wired together in a shallow, fully connected neural network to combine the information learned in two separate branches to make a final prediction for drug response. The interpretability of the model's prediction comes from the genotype side of the model, to which we imposed a structural restriction to make it mirror a hierarchical organization of cell functions and becomes biologically meaningful. We used the gene ontology as the hierarchy. Each cellular function or pathway in the hierarchy is assigned with a fixed number of neurons. And instead of fully connecting all neurons between layers, we determine the connectivity between neurons representing each pathway based on the hierarchical relationship between the pathways as defined in the ontology, gene ontology. Have this framework implemented, we trained a model using about 500,000 data points for drug response between 1235 cancer cell lines and 684 drugs. The training data is extracted from the combination of the databases curated in Genomics of Drug Sensitivity in Cancer, aka GDSC, and Cancer Therapeutics Response Portal, aka CTRP version 2. The drug response is measured using the area under the dose response curve, which I will refer to as AUC from now on. The example of the training data is shown on the slide. Each cell line is translated into a binary vector representing the mutation status of 3008 most frequently mutated genes across all cell lines. Each SMILES representation of the drug is translated into Morgan fingerprint, a binary vector whose length is 2048. Then the pair of the binary vectors are fed into the model with the drug response AUC values. Then we evaluated the predicted performance of the drug cell model. The accuracy of our prediction was measured using the Spearman rho between the actual drug response AUC and the predicted AUC, and it was 0.8, as you can see in the first plot. You can also see that the distribution of the model's prediction has a good linear relationship with that of actual drug response. The second waterfall plot shows the list of drugs sorted based on the per drug performance. The red area is the drugs with the Spearman rho bigger than 0.5, which are used for most downstream analysis moving forward. And instead shows the top 10 drugs with the drug cells high prediction performance, and those cover drugs in different target classes. When I compared drug cells performance to 3 other baseline models, ElasticNet, which is a linear regression model, black box model with a matched number of neurons, and another black box model using a tissue type as additional feature. You can see the drug cell outperforms the other models for many drugs in the scatter plot. In each plot, each data points represent the individual drug, and the points above the diagonal line are the drugs that the drug cell could make more accurate prediction than the other model in comparison. After evaluating the accuracy of the drug cells prediction as a proof of concept, we analyzed the drug cells prediction and interpretation for a microtubule stabilizer, paclitaxel. The predicted performance of drug cell for paclitaxel was 0.79 in Spearman rho. Then the question now is how the drug cell model made the predictions for paclitaxel, and in other words, what is the interpretation of the model's prediction? To answer to that question, we used the metric defined in the previous publication of the Edgar lab called relative local improvement of predicted power, which I will call OLIP for the rest of my presentation. Olive score compares the predictive power of the neuron values assigned to a pathway to that of the concatenation of the neuron values assigned to each children. The parent-children relationship is defined in the genontology again. To calculate the Olive scores for one pathway, I first trained a linear model using the neuron values assigned to the pathway. Then I collect the neuron values assigned to all of its children pathways and concatenate them, and then train another linear model using the concatenation. Then the Olive score is defined as the ratio of Spearman row of the parent linear model to that of children linear model. Olive score larger than 1 reflects the state of the parent pathway represented by its neuron values, has more predictive power than for the drug response than the mere concatenation of the state of its children. And it indicates the importance of the parent pathway in the drug cells learning. So going back to paclitaxel, I calculated the Olive scores for all pathways in our genontology and sorted the pathway based on the Olive scores. The noticeable observation that we made is that there were many metabolic pathways within the top 5% of the pathways. So we examined the connection between paclitaxel and the metabolic pathway by treating a 4 to 7 lung cancer cell line with paclitaxel and a glycolysis inhibitor to deoxyglucose and compared the cell viability after treatment. The white box is the cell viability distribution when only the vehicle is injected to the cell line, and blue box is when treated only with the paclitaxel, and gray box is cells treated with the to deoxyglucose, and finally the darker blue is the cells treated with the combination of paclitaxel and to deoxyglucose. The plot shows that the more cancer cells died when treated with the combination of both treatments than when treated with an individual drug, indicating the synergy between paclitaxel and to deoxyglucose. Based on the observation in the paclitaxel analysis, we concluded that the genotype branch of the model learns synergistic targets of the drugs in the examination. Then we dived into the question why the model is learning about synergistic targets of the drugs in the genotype branch. To find the answer to that question, we examined the inner working of the drug cell model more closely. We first looked at the what the drug embeddings that drug cell learned recapitulate. We performed the PCA on the top level of drug embeddings and visually inspected the data points in two-dimensional space. And then we observed that the drugs that are in the same target classes are well clustered together in that space. This means that the drug embeddings are learning about the mechanism of actions of each drug. Now moved on to the genotype branch, we try to answer to the question, how does a drug cell learn about the synergistic drug target? Considering the synergistic relationship between the drugs and the high Olive score pathways, we concluded that the model's inner working mirrors very well the parallel pathway inhibition theory of synergy. The parallel pathway inhibition theory holds two drugs are synergistic when they inhibit separate pathways that regulate a common essential function. So we hypothesized the biological activity of primary drug is learned by the drug embedding branch, and the parallel pathways are learned by the genotype embedding branch, where the secondary drug's activity is simulated by existing loss of function mutations in each cell. To systematically validate the drug cell's findings about the synergistic targets made using Olive score, we did the following analysis using a public database called DIP Synergy, which collects synergy scores of drug combinations between 32 drugs. For 25 primary drugs, both existing in DIP Synergy and our training data, we ran the Olive score analysis. And using the top 5 and bottom 5 Olive pathways, we for each primary drug, we identified the potentially synergistic and non-synergistic secondary drugs and compiled the two sets of drug combinations. Our expectation was that the drug combinations compiled using the top 5 OLIP pathways would have higher synergy scores than the one compiled using the bottom 5 OLIP pathways. And as we expected, we observed that the distribution of the synergy scores of the drug combinations compiled using the top 5 OLIP pathways are shifted up compared to bottom 5, and all the rest combinations are existing in the deep synergy databases with the statistical significance. There are a few more experimental validation for the drug cells finding conducted using the cell lines, but in the interest of time, I will skip them in this presentation. Please find more information about those validation studies in our paper. With that said, I'm going to shift gears towards the very important evaluation for the model trained using the cell lines, which is whether the model can be generalized to other preclinical models or even patient data. To perform such a validation, I assessed the data in PDX Encyclopedia, where there are 399 PDX tumors of various tissue types, screened against 40 monotherapies and 27 combination therapies. First, we directly used the drug cell model, again pre-trained using the cell line data, and predicted the response for the PDX models to the monotherapies. Since the drug response is measured using progression-free survival in the PDX data, which is different from our measurement, which was AUC, we first stratified the PDX models into drug cell positive group that are sensitive to the treatment and drug cell negative groups that are resistant to the monotherapy based on the drug cells prediction. The binarization is done using the median predicted drug response AUC value. Then I compare the median progression-free survival rate between the two groups. The drug cell positive group had a longer median progression-free survival rate than the negative group. And when we also compare the distribution of the progression-free survival rate between two groups using log-ranked tests, the two distributions were different with the statistical significance. Then we went ahead and evaluated the drug cell's ability to suggest effective drug combinations for the PDX models. To do so, we performed the OLIP score analysis for 6 primary drugs that are included in both PDX data for combination therapies in our training data, and evaluated the efficacy of the suggested drug combinations by the drug cell. To determine the threshold for the percentage of the top pathways suggesting synergistic drug combinations, we performed a RC curve analysis with the sweeping the portion of pathways to use for the suggestion. As a result, top 20% of the OLIP pathways were used as synergistic targets, and all the rest pathways were considered as non-synergistic targets. The contingency table counts the number of the effective and ineffective drug combinations defined by our model and in the data. The right plot shows an example of effective combinations predicted by drug cell, BKM120 and encrofenib, BKM20 and encrofenib, and Abraxane and gemcitabine. You can see the progression survival rates were improved for PDX models that received both treatments than the PDX models that received just one of the treatments. On the other hand, for the drug pairs predicted as ineffective by drug cell, for example, paclitaxel and LCL161 and INC280 and trastuzumab, we found that the progression free survival rate didn't really differ between the PDX model that received both treatments and those that received just one treatment. Finally, we sought to evaluate whether DRUXA can be used clinically to stratify the patient tumors into responsive and non-responsive groups. Using a breast cancer cohort in AACR Genie project containing 221 ER-positive metastatic breast cancer patients who had undergone multiple rounds of therapy, including mTOR inhibitor and CDK4/6 inhibitor. Treatment outcome was measured using overall survival rate in this data set. Using the pre-trained DRUXA model, we predicted sensitivity of breast cancer patient to either mTOR inhibitor or CDK4/6 inhibitor, and stratified the patient into sensitive, DRUXA positive, and resistant, DRUXA negative groups, again, using the median predicted AUC as a threshold. We then compared the distribution of the overall survival rates of the sensitive and resistant patient population that DRUXA predicted. As you can see in the plot, the predicted sensitive patient group has statistically significant longer survivor than the resistant group. When we looked at the specific genetic alterations within the sensitive group and resistant patient population predicted by DRUXA, the sensitive group is much more likely to harbor AKT1 mutations than the resistant group. However, the resistant group had mutations in various genes previously known to be associated with the drug resistant, including ESR1 and then RB1 and PTEN. And this suggests that the drug cells stratified the resistant group based on the complex pattern of mutations. And interestingly, for the sensitive group, AKT mutation status was not the best predictive of the therapeutic response. If you look at the plot on the right, AKT mutant patients actually had shorter median survivor rate, and the difference between the overall distribution was not statistically significant. This implies that DRUXA's prediction for the sensitive group is also based on the combination of mutations rather than the status of a single gene. So this analysis illustrates how DRUXA model can be used to effectively guide clinical treatment decisions with significantly greater precision than the single gene marker studies. And I also performed the olive score analysis for mTOR inhibitor and CDK4/6 inhibitors. The analysis suggested that the two treatments are potentially synergistic pairs, as they prioritize CDK activity for mTOR inhibitor and TOR signaling pathway for CDK4/6 inhibitor. But this Genie data doesn't have a proper drug response information for the combination of two. We examined their synergy using the PDX model data that I previously showed. In the right plot, you can see the progression survival rate of the PDX models improve when treated with the combination of mTOR and CDK4/6 inhibitors compared to when treated only with each treatment. With all that said, let me summarize what I have presented so far. Today, I introduced an interpretable deep learning model for drug response prediction and have shown that its predictive performance was not compromised too much to gain the interpretability. And the interpretation of the model highlights the synergistic drug combinations, and relevant findings are validated using the preclinical models and patient population data as well, indicating the model's generalizability. So I hope I convinced you that the DRUXA has great potential to aid the improvement of clinical trials. And where do we go from here? I will quickly cover a few of the future directions. We can first think about to improve the DRUXA model. We can first think about the incorporation of the other features of the cells and drugs, including, for example, for cell genotypes, we can consider expression profiles or epigenetic states or microenvironmental influences. And definitely secondly, we can think about improving the hierarchy of the cell functions embedded in the drug cell to make the interpretation more seamless and more biologically meaningful. Lastly, drug cell can only learn for the drugs with the known chemical structures, but the framework can be adapted and modified to learn about the therapies without known chemical structure as well. But that's going to be more of a longer term open question. Lastly, I will acknowledge everyone in the IDECA lab, especially Trey Idecker, my postdoc supervisor, and Brent Quenzi, who is the first author of the drug cell paper. And I thank our funding agencies. And with that, I will take questions.
Excellent. Thank you very much, Jisoo, for an outstanding talk on knowledge-guided machine learning applied to translational biology from cell lines to patients. There was one question that came up in Slido. It was relatively early, and it says the predicted drug response in Spearman, is Spearman correlated? I think this was the original development of drug cell, but the model appears to overestimate the effect of drugs within AUC, I guess I measured AUC around the 0. Any explanation for this bias?
You mean here?
I perceive so, yes. I
see. Yeah, it's one of the things that we have to also think moving forward to further improve the model's predictive performance, which is that the data, one thing that I should mention, I should tell you about the data itself is that in the data, the training data is pretty imbalanced, that there are more resistant drug response data than the sensitive drug response data. We have discussed a lot about how to resolve the imbalance in the between the sensitive and then the resistant drug response data. But yes, there are, so for the it's well thought and considered that for the sensitive cell response to the drugs, it's a harder problem to predict. That's why it's actually yeah, I'm not sure that it answered that question completely.
Okay, well, no, I think that's good. Yeah. All right, thank you again. We're going to move on for the sake of time. Our next speaker is one of my colleagues, Peter Cassin from the University of Virginia. I've known Peter for a number of years, and he's going to talk with us about better inference through chemistry, knowledge-guided inference of biomolecular kinetics. And so, Peter, if you can pull up your slides.
Great, thank you. It's a pleasure to participate in this workshop. So what I want to talk about is a set of approaches to understanding the kinetics of macromolecular machines using sparse experimental data and two different types of sparse experimental data. One is where we have structurally specific data. So on, for instance, proteins, if we have labels at sites we know on those proteins, and we have then information about how those labels interact with each other and give us distance information and distance constraints and how they evolve over time. And that yields one set of methods to try to understand how macromolecules have structural and kinetic diversity. The other is a class of more challenging data that I would say are structurally indirect, where we have reporters on a biological process. And the example that I'll use, and we'll go into more detail, is for looking at viral entry, where we can measure this on a single virus level using fluorescence dequenching. And so then we get fluorescence signals that reflect a state change in the biological process, but are not trivially assignable to specific molecular states. And we're working on actively developing methods for kinetic refinement of these using statistical learning, also using some physics-based methods. And so there's, for both of these, there's a lot of interplay between the sort of philosophically related areas of statistical learning and statistical physics. What I want to share with is our current state, which has maybe somewhat more mature methods for statistical physics-based inference than statistical learning-based inference. But we believe very strongly that these two work together. And I also want to explain some of the challenging nature of the problems. So why we care about this is to try to infer kinetics and mechanism across scales. And so what you see on the left is a movie of the kinds of experiments we do on single virus entry. These spots are each individual influenza viral particles. And as they, in the movie I played, as they got brighter, those were the particles fusing and becoming infection competent to insert their genome into a cell. And at a much more detailed level, we have information from, say, cryo-electron microscopy on the upper right that gives us the architecture of these viral particles, and then very detailed molecular models, as are shown on the lower right, that are models that can integrate some structural data, but nonetheless have a very substantial gap in spatial and temporal scales between these and the sorts of inference of kinetic models we can do based on molecular dynamics trajectories and the sorts of inference we can do on kinetic models based on viral fusion trajectories. Why we care about this is a combination of mechanistic insight and therapeutic design, that we have data showing that antibodies to influenza and other viruses can bind to essentially the same place on the viral protein, yet work differently. And this is perhaps not surprising, because these viral spike glycoproteins that I think you'll also hear a lot about in the next talk are proteins that act by taking on a number of different shapes and by mediating multiple steps of attachment and entry of the virus, and therefore looking at it in a purely static model to try to infer mechanism and also try to optimize therapy ends up being likely insufficient. And so the first problem that I want to talk about in this regard is a problem on the way to kinetics, and I'll explain a little bit where we want to go, but of structural ensembles that both for viral glycoproteins and for many other flexible proteins in biology, that we can sometimes resolve one or a few states at high resolution with structural information, but the full ensemble of states and often the intermediates contain much of the important mechanistic information for figuring out how these function and also how to engineer them. And however, for getting at structural ensembles, we often run into a problem of sparse experimental data, that the experimental techniques that report on structural ensembles tend to yield a small number of degrees of freedom relative to the number of constraints required to resolve molecular structure, yet they can give distributional quantities rather than just mean values for these degrees of freedom. And so that's shown on the left with some single molecule FRET data from the laboratory of my colleague, Walter Mothis. What we want to do is be able to take these sparse data and use them to infer the structural ensembles of, or infer as much information as we can about the structural ensembles of the molecules that they're reporting on. And one of the goals we have is transferability. That if we have extensive information about a molecule in one environment or under one set of conditions, can we use sparse and potentially expensive to acquire data that are many fewer data to then transfer our knowledge about the structural ensemble in one environment to another environment. And from statistical physics, if our if our methods for adapting our structural models from one environment to another satisfy maximum entropy properties, then that is then the least perturbing transformation of the model from one to another, and thus represents the best we can do to understand the protein in its new environment. So that's some of the motivation. One of the methods that we developed, and again, this is more a physics-based inference, is to be able to estimate these heterogeneous conformational states based on sparse distributional data. One of the challenges is just refining this in a stable manner when the data are relatively heterogeneous, that many of the prior approaches work well when you have probability distributions that are continuous or near Gaussian. And so we developed an approach that is very similar to stochastic image formation, that essentially takes an initial model of the structural ensemble of the protein. This model can be a bad model, and then draws random samples from that and random samples from all our experimental data distributions, and essentially refines each sampled conformation against a distribution that looks like a delta function that's a random sample from the target from each of the target distributions, and then collects all of those and uses that ensemble to update our estimate of the structural ensemble of the protein of interest. And we can do this fairly well, and indeed outperform for these challenging distributions prior state-of-the-art approaches. And so this then gives us the tool to do what it says are the interesting pieces. And one is, again, utilizing some properties of statistical physics. If the experimental data are acquired separately in different experiments, they then contain no information about the correlation between them. And most approaches in the field have treated those then as independent, and the joint distribution as simply the convolution of the individual distributions. Whereas we know that in many cases, if you're measuring a protein that's flexible and moving in different ways, that there has to be correlation between the different measurements. And one can illustrate this with a very simple toy model of an alternating transporter shown on the left that can take two conformations and the sort of intermediates between those, but where the two measurements are very clearly coupled, and where if one just assumes that you get the two individual distributions or the joint distribution is the convolution of the two individual distributions, you get something very different from the true analytical distribution shown in panel C here. What we can do is to essentially perform physics-informed inference that estimating the non-equilibrium work required to restrain this, the system to any point in this joint distribution yields the probability of that point in the joint distribution when averaged over many trials. And so again, via sort of a stochastic approach, we can build up the correct joint distribution after a large number of runs. And that's shown here. And then we can apply this to a flexible protein such as syntaxin, shown on the right, which is a neuronal protein involved in neurotransmission, and yield elements of the structural joint distribution, both states that would be, if we don't know any of this correlation information between the experiments, we come up with some states for the protein that are actually physically disallowed, and certain other states in this joint distribution are much, much more likely. And so we can greatly improve these estimates. What we really want to do with this is to have a statistical inference cycle for refining structural ensembles of flexible proteins, where we know how to best incorporate experimental data and then can best choose the experiments, given that these experiments are relatively low throughput and relatively expensive to perform. And so the next component, I've told you about some refinement approaches, is to ask, can we identify the most informative experiments to perform to further refine conformational ensembles? And so for this, we took a relatively, in the information theory community, a relatively well-identified quantity, which is a maximum relevance minimal redundancy quantity between different potential labeling sites on a protein. And we showed that even given very noisy starting estimates for conformational models, we could select experiments, use the resulting data to refine those conformational ensembles, and substantially outperform experimental selection by a trained spectroscopist, in large part because our estimates both could eliminate redundancy better, and we could also know when to take certain risks in experiment selection much better than a spectroscopist who ended up being much more conservative in those cases. And so then this gives us a way to complete that cycle and have methods for inferring the probability distribution of structural ensembles given experimental data, and methods for selecting experiments to go on to further refine these structural models. The next cases, of course, will be to use many of these experiments, but not all, also yield dynamic information. And that's going to require a little bit more statistical inference. That's something we're working on very actively right now. I'd also say that, I've shown some non-viral examples for this. We're also working on HIV glycoprotein and other viral glycoprotein structural ensembles, and can match experimental data very well with that. The challenge for these larger scale non-structurally specific experiments is that the states are highly degenerate, that these fluorescence dequenching data that I've shown you, if we look at a single virus trace on the upper right, it's essentially a step function. And so we can measure a state change from not entry competent to entry competent, and we can build this up into a cumulative distribution function over many trials. But there are many molecular states and indeed many, often many kinetically important states that all have this initial emission value as opposed to the final emission value. And this degeneracy makes it very difficult, as I'll sort of briefly show you in the final few minutes, to train a lot of traditional statistical inference models. What we've done in the past is traditional sort of fits where one can postulate a kinetic model that corresponds to a series of differential equations and perform inference-based fits of these models. But this is really very, very driven by specific hypotheses. This is for Zika virus, and can indeed yield substantial insight from this. But we'd like something more general. And also, furthermore, for Zika, we can change a parameter and see differences in a number of rates, but the functional form, this number of intermediates in the existence of an off-pathway state, which we'd like to exploit for inhibition of viral entry, is all the same. However, when we look at influenza, if we change some of these parameters, as you see on the left, the functional form actually changes, and we can quantify this using methods that were developed for single molecule enzymology. And we can see this change both when we change temperature and when we change other physical parameters in the experiment. And what we'd really like is an inference framework to be able to extract the dependence on these physical parameters and encode what we know about how model families should depend on these parameters in a principled manner and in as general a manner as possible. This is challenging. I sort of show you the success here. This is very much work in progress. That hidden Markov models with a number of constraints can fit some of these very well, but others, because of the state degeneracy, it becomes very difficult to converge traditional models, and a number of the sort of encoding prior knowledge and regularization approaches become very important. What we're working on is one step beyond that, to be able to properly encode shared parameters across the model families. And what differs from sort of off-the-shelf models here is that the time series update step reflects the chemistry, that we know how update steps should reflect, say, temperature or some of these other things. And so it's not just a linear dependence. There are nonlinear dependencies in the update step that make life a little more complicated, but very interesting. So I'll wrap up there. I'd say we're actively using, particularly where there are sparse data, a combination of statistical physics and statistical inference. State degeneracy for these sorts of problems is extremely challenging. And the thing that I find most exciting is being able to encode chemical knowledge within an inference framework. Pardon me. With that, I'll just thank the members of my team, our funders, and all our collaborators. And if there's any time, take questions.
Excellent. Thank you very much, Peter. I'm going to take session organizer's prerogative, and I'm going to ask you a question. You mentioned multiple times the challenge of degenerate states and sparse data. The question that was on my mind is how much of this is just the essence of protein dynamics that you are studying, and how much of it are a function of the probes that you're using to assess those states? In other words, if you had infinite knowledge about everything, with atomic resolution, what the protein is doing, would that resolve the sparsity issue? Or simply are the biophysics of these proteins such that they're just these rare occupancies that you'd never catch them no matter how good your detection reagents are?
If one had the ability to, measure everything in high time resolution, we should be able to resolve these. I think the challenge is that the nature of the measurements, and even some of these measurements are perturbing by themselves, is that they can't resolve particularly transient states. And then also some things are degenerate to the accessible probes.
Does that open the door then that better probes or better measurement technologies may eventually resolve that challenge? Or have you hit a type of measurement limit for where some of those are? In other words, how fast you can snap the camera, or how many times you can look at a fluorophore before it starts bleaching?
I think there will be some fundamental limits. I think what we can do is also increase our ability, particularly since for many of these we actually control the experiment design as well, increase our ability to do experiment selection. And so if we can use the models and the uncertainty in the models to guide probe design and experiment design, we can take this farther. There will be some fundamental limits, but we're not quite there.
Okay. All right. Thank you again, Peter, for a nice talk. We're going to move on to our final speaker, Brian Heave from Stanford University. He's a Stanford Science Fellow there. But what I think he's going to talk about is work that he did as a graduate student at MIT. I've heard about this work around the way, so I'm very interested to hear Brian's talk on it. And so the title of his talk is Predicting Evolution with Protein Language Models. Brian, the floor is yours. All you need to do is unmute now.
Okay, sorry, there's a problem with my Zoom. Am I muted? Can you hear me?
You're fine now, and I can see your slides.
Awesome. Cool. Great. It's great to be here. Thanks, Kevin. My name is Brian He, and today I'm going to be talking about how to leverage a class of algorithms called neural language models. And as the name kind of suggests, these were originally developed in the context of natural language processing. But instead, I'll be talking about how to apply them to a completely different set of applications, in this case, predicting and modeling evolution. And it turns out that these language models can be quite powerful in doing so. So this is joint work with some great co-authors, and was done at two different institutions, MIT, where I did my graduate work, and Stanford, where I'm currently a science fellow. All right, so let's get started. To begin, I just want to motivate this talk with this really high-level question, which is this pretty fundamental question in biology, which is, how predictable is evolution? So on one end of the spectrum is this thought experiment by Gould, where you rerun the evolutionary process, starting with the universal common ancestor again, but you rerun that process for all life on Earth, and say Gould hypothesized that evolution is completely determined by chance, and if you rerun this process, you'd get these fantastic new organisms with different kinds of life that don't resemble anything that we see on Earth today. But on the other end of the spectrum is this hypothesis by Morris, where actually evolution is so constrained by intrinsic rules that if you reran that evolutionary experiment, you would get back humans or a species very similar to humans. And so two ends of the spectrum, where on this end of the spectrum, there are intrinsic rules that are possible to learn, and in doing so, to be able to predict evolution. And so this is kind of the high level framing of this talk. And so for the majority of the talk, I'll be describing some recent work on trying to predict and model the evolution of viral surface proteins. And then towards the tail end of the talk, I'll kind of devote a little bit of time to some unpublished work on how to extend some of these ideas beyond viruses to generic proteins and proteins evolving over much longer evolutionary timescales. But to begin, I'll be talking about predicting viral evolution, and I think the practical application of this is pretty straightforward, given that we're in the midst of a global pandemic, and in particular a particularly kind of bad or challenging problem in developing antivirals and vaccines is this problem of viral escape. So say you have initial immune recognition of this viral surface protein, and this antibody will bind and neutralize the virus, and this host will have immunity to the virus. But what viruses can do is that over many generations, they can mutate their surface proteins so that, say, this neutralizing antibody no longer binds, and then this viral particle is then free to infect the host again. And this is the problem of immune escape. And so viral escape is a pretty big problem. It's the reason why we don't have a universal vaccine for influenza, why you need a yearly flu shot. It's the reason why we don't have any effective vaccine at all for HIV, despite decades of intense research effort. And there are questions about the durability of vaccine protection for SARS-CoV-2. At the moment, it seems that the vaccine-induced immunity is the virus. It's holding up well against the current variants of the virus. But as SARS-CoV-2 continues to evolve, there's definitely questions about whether or not it will start to escape the immunity that's generated by even the best vaccines we have today. So how do we model viral escape? Using kind of algorithmic techniques. An initial kind of key insight that we had is that small changes can have big semantic effects. And so what do I mean by this? So say you have this sentence, the boy pats the dog, and I can actually make a single character change to the sentence that completely alters its meaning. So this is a very different sentence, but in terms of the characters, I just mutated a single character, a P to an E in this case. And actually, a similar phenomenon happens in the case of viral escape. So actually, if you mutate, say, this histidine to a serine on this influenza hemagglutinin surface protein, then this broadly neutralizing antibody no longer binds, and this surface protein can, or this viral particle, if it had this mutation, could, say, escape from this neutralizing antibody. And so an initial kind of question that we had is there something kind of related between the way that a virus can escape the immune system and the way we can model semantic change using machine learning? So to make this a little more concrete, this is how we're kind of thinking about the connection between kind of the language of viral escape. So say you have some initial protein and it has some initial antibody that's recognizing it. So if I'm this virus and I want to escape from this antibody pressure, what do I want to do? So number one, I don't want to mutate myself so that I die, or that the protein misfolds, or I lose infectivity. So I want any mutation that I make to my surface proteins to preserve some notion of acceptability or validity or grammaticality. But what I also don't want to do is I don't want to mutate my surface protein so that it just looks pretty much the same to the immune system and I still get recognized by this antibody. So kind of a functionally neutral or functionally maybe synonymous mutation. I also want to avoid this regime. Instead, what I want to do is I want to preserve my ability to infect cells but I want to substantially alter my surface protein so that I can kind of escape from this antibody pressure. So this is how we're thinking about viral escape, kind of preserving a degree of acceptability or viability or grammaticality, but also being substantially altered in some way, or substantial, maybe semantic change. And to kind of formalize this a little more, this is the kind of key kind of word game that we introduce to kind of model viral escape. And it's what we call a constrained semantic change search, or CSCS. And so it's a pretty simple kind of word game. You're given a sequence of tokens from some language, and your goal is to find the single token change that, number one, induces the largest semantic change to that sentence, but these changes have to be constrained. They have to fit within some set of rules or a grammar of this language. And this game is given in generality, so this could be a sequence of words from an English sentence, or a sequence of amino acid residues from a viral protein. How do we implement this CSCS model? Well, we can use these algorithms called language models, and at a high level, what they do is they'll assign, in this masked language modeling case, they'll assign a probability to some token appearing within some sequence context. So how probable is some amino acid residue appearing at this position given the rest of the amino acids in that sequence? And when you train this language model on, say, thousands of infectious viral sequences, the output probability will begin to model something about the validity of mutation or the grammaticality of a given mutation. So that's the first part of CSCS, right? We want to learn the grammar of the language. But the second part is we also want mutations that induce high semantic change. So to model semantic change, we leverage this idea from, again, borrowed from kind of natural processing and Elmo and these models, where you can look at the distance in the semantic embedding space. So this is the output of one of the hidden layers of the language model, and it's a continuous embedding, and kind of sequences that are closer in this semantic embedding space, say via the Euclidean distance, are also more semantically similar. And as that distance increases, your semantic dissimilarity between those two sequences also increases. Great, so now we have a way to model grammaticality and semantic change. And our CSCS objective is actually quite simple. So we rank over all possible single token mutations to the sequence. We rank them by the grammaticality, and then we rank them by semantic change. And then we just acquire based on high grammaticality and high semantic change. Of course, you can imagine other potential objectives, but this is the objective that we use. It's a pretty simple one. So this is a bit abstract, but just to root this in some intuition, we actually trained a language model on news headlines. It's a word level language model. And so here are some real CSCS changes. So for example, we have this headline, Australian dead in Bali. Now, if we ask the language model, what is the semantically closest sequence that's just one word away, we get this mutation, Aussie dead in Bali. So this is pretty synonymous with the original headline. But when we ask for the CSCS proposed change, we get Australian ballet in Bali. And so you can imagine that this is a valid headline. You could see this appearing, Australian ballet in Bali. But all of a sudden, it's a lot more positive. I guess depending on your opinion of ballet from the original headline. Some other examples, blast off of Apollo 8 gets turned into blast victims of Apollo 8, all of a sudden a lot more negative. This is another kind of strange headline to begin with. Excuse me, you left a gorilla suit on the bus, gets turned into excuse me, you left a gorilla killer on the bus. So a lot more negative and more strange. You can kind of do this all day, but I'll give one more example. Wine growers revel in good season gets turned into wine growers revel in flu season. And so when we kind of stepped back and thought about this, what we're actually doing is quite similar to these games, Mad Libs or Apples to Apples or Cards Against Humanity, if you're familiar with these games. And the reason why we find these games so, humorous is that actually what they're doing is they're enacting this constrained semantic change search. So the rules of the game constrain the word choices that you provide the game to be grammatically correct. And the reason why we find these games amusing or funny is this, semantically the semantically jarring at times word choices that we make when we play these games. So we kind of stumbled on a kind of formalization of Mad Libs or Cards Against Humanity. But this is, somewhat a toy example just for intuition. What we were actually interested in is, applying this to see if we can actually model viral escape. And we can actually just train a language model instead of on sequences of words. We can just train, using the same techniques, a language model on sequences of amino acid residues. And so when we train a language model, say on about 50,000 flu hemagglutinin sequences in the influenza research database, and we visualize that language model embedding, it's a very high dimensional vector in 2 dimensions that try to capture as much of the variation in that high dimensional embedding space as possible. And we plot them in 2 dimensions. You can see that each point here corresponds to a sequence. You can see that the sequences start to cluster according to their known influenza subtype and their host species. So what this is showing is that these language models are learning an internal representation that captures something about the antigenic similarity between these sequences. It's not learning something completely random. It's learning these kind of antigenic patterns consistent with our hypothesis. Again, if we train a language model on the HIV envelope surface protein or coronavirus spike, homologous sequences. In the envelope case, again, these sequences will cluster by subtype. And in the coronavirus spike case, these well-known human coronaviruses actually cluster with the hypothesized and zoonotic origin of these human coronaviruses. So SARS-CoV-1 clusters with bat and civet, MERS-CoV clusters with camel, and then SARS-CoV-2 clusters with these bat and pangolin sequences consistent with their kind of known or hypothesized zoonotic origin. Right. So we know that these language models are learning something about kind of the antigenic distribution of viral evolution, but can this actually predict escape? So to leverage, to kind of validate a model for predicting escape, we can leverage these deep mutational scan data sets. In this case, this is from a paper by Jesse Bloom's group at Fred Hutch, where you can make all single amino acid residue changes to hemagglutinin, and then grow up this virus in the presence of neutralizing antibody or human sera, and then infect cells, sequence these the viruses that make it inside these cells, and then compare to a control setting to find differentially selected mutations that will escape from this immune pressure. And in this way, it's kind of a very high throughput way to identify all single amino acid escape mutations to a viral surface protein. And rather strikingly, when we plot all possible single amino acid residue changes to a surface protein, which is each dot here in this 2-dimensional plot corresponds to a single mutation. And when we plot them according to grammaticality on the x-axis and semantic change on the y-axis, and then we also overlay the dots with the known escape mutations from that deep mutational scan, you can see that the escape mutations kind of exclusively cluster in this high grammaticality and high semantic change quadrant of this kind of 2-dimensional grid. And if we quantify this, we can also demonstrate that this escape enrichment is statistically significant. And so this is a great validation that how we're thinking about grammaticality and semantic change is actually aligning with what we see in this kind of in vitro model. And I just like to emphasize that this language model that was used to rank grammaticality and semantic change was trained on just on hemagglutinin sequences we did not provide any explicit information on escape mutations. It just had to learn these patterns from the data. If you visualize this escape enrichment on the three-dimensional structure of these service proteins, you see that there's a lot more escape potential in the head of influenza hemagglutinin, for example, than the stalk region. And this is consistent with the known kind of escape strategy of influenza. And for SARS-CoV-2 spike, if you train a spike language model, we again see significant escape potential in the receptor binding domain and the N-terminal domain. And when we made these predictions in, I guess, July 2020, it was kind of heartening to see that in vitro models, these two domains were the ones that were kind of showing up for escape mutations, in particular the NTD deletions and kind of loop insertions in the NTD are kind of highly suggestive of escape from antibody. So you're probably wondering if this model would be predictive of emerging SARS-CoV-2 variants, and indeed we did do this analysis in a follow-up study. So in this follow-up study led by Cyrus Mayer at VIR, we can actually show that spike language model features are predictive of mutations that arise in circulating coronavirus variants up to even 4 months in advance with a high AUC of around 0.8. And so this is kind of an additional validation that the evolutionary information that's picked up by these language models, and in particular language model grammaticality, can be predictive of mutations that preserve viral fitness even across different waves of the coronavirus pandemic. In particular, especially this one mutation, S494P, that gets highlighted by our language model and other evolutionary features, does have experimental evidence of reducing sensitivity to these neutralizing antibodies that have emergency use approval for SARS-CoV-2. And so this is experimental confirmation that some of these escape mutations are in fact reducing sensitivity to antibody therapeutics. Right. So that was how we can kind of apply language models to viral evolution. In the last, say, maybe like 3 to 5 minutes of this talk, I'll just briefly go over how these ideas can expand to incorporate more than viral evolution, but generic proteins and proteins that are evolving over much longer timescales than SARS-CoV-2 spike or even hemagglutinin. And so we know that these language models can predict local evolution or single amino acid changes. But a big open question is, what about mutations that are more complicated, like more mutations due to recombination or more than single residue mutations? So a key insight that we had is that you can actually gain some insight into the global landscape of evolution by composing a bunch of these local predictions together. And the kind of agglomeration of these local predictions gives you an approximate insight into the global kind of landscape of evolution. Conceptually, this is kind of similar to RNA velocity, if you're familiar with that. If not, it's not a big deal, but RNA velocity, you just try to predict locally the transcriptome that is nearest to you in the future state via splicing kinetics. And then these local transcriptomic predictions can be assembled across an entire trajectory to gain insight into the full directionality of, say, cellular development. From an evolutionary perspective, this is also similar to ideas of the fitness landscape from Sewell Wright and others, where a protein is just making locally optimal improvements as it evolves, and in doing so, it's exploring it's doing hill climbing in this fitness landscape. So in particular, we're interested in these universal protein language models where you train a very large neural network language model on all sequences, say in UNIREF 50. And in doing so, we're hoping that this is a language model by Rives et al. From Facebook AI research. And when you train a language model on 3 million sequences, hopefully it's learning something more general about the evolutionary rules involved in what makes, say, a valid protein. So when we apply this language model to kind of stitch together the flow of evolution through this landscape of, in this case, influenza nucleoprotein, what we find is, these aren't kind of stream plots of wind moving through a 2D landscape, but in actuality, this is kind of like the flow of evolution across this mutational landscape of nucleoprotein. And when we visualize this, we can see that the actual flow of evolution, as predicted by the language model, corresponds to the known temporal evolution of the nucleoprotein. And we can predict the roots of the major pandemics of influenza, and in particular, this one well-established phylogenetic path of nucleoprotein does indeed move according to predicted directionality of evolution by these language models. So you can kind of visualize the flow of evolution occurring across this protein landscape. Because we're using these universal protein language models, we can apply this beyond viral evolution. So this is what the kind of flow of evolution looks like for cytochrome C. And what we've found pretty interesting is, the predicted pseudo time, if you order based on this flow, recapitulates the ordering of these taxonomic categories in the fossil record. So it's kind of nice validation that the ordering information from this velocity of evolution is being recovered. And we can also show that this extends to even kind of the longest evolutionary trajectories spanning all 3 domains of life, bacteria, archaea, and eukaryota. And we can also similarly kind of predict the directionality of evolution and model this flow using these universal protein language models. So to go back to that initial question, there's probably a lot in evolution that's just due to chance. I'm not claiming that it's completely constrained. There's a lot of environmental factors involved. But as Russell Schwartz and others have so clearly demonstrated during this session, there is also a lot of inherent rules that are constraining evolution, and that's what makes these language models probably particularly powerful in being able to extract these rules just directly from large corpuses of sequences. And so it's an exciting new area, the ability to maybe provide some insight in predicting evolution just from sequence data alone. There's a lot that I omitted just due to the sake of time. So these are the references describing this work in more detail if you're interested. And I'd like to thank you all for your attention. This work is done with an amazing group of co-authors. Ellen was a collaborator on the viral project in collaboration or under the supervision of Brian Bryson and Bonnie Berger, all at MIT. And then the velocity work is done with Kevin Yang at Microsoft Research and Peter Kim at Stanford. So thank you.
Terrific. Thank you very much, Brian, for an excellent talk. We have a couple of questions on Slido. For the sake of time, I'm going to try to glom them together into one question. And it relates to porting over this idea of grammaticality to viral sequences. And kind of the related question is you're substituting letters, and in your diagrams, you appear to be substituting amino acids. But in reality, the changes that happen in virus sequences happen at the nucleotide level, the base level. So you have, I don't know how would I describe, like a Hamming distance sort of thing to take into consideration about how many changes you would need to swap certain amino acids. So could you give a little bit more detail on how those are implemented in your the language model?
Yeah, what's great about these language models is that they're general. So you can use any kind of alphabet in your language. So in our particular language model, we give it an amino acid alphabet, and we just break the language, the sequence up according to amino acids. But you could just as easily train a nucleotide level language model where you just use 4 characters in the alphabet. You could also just easily train a larger scale language model, or maybe 5 MERS or something are your your alphabet or the level of genes, a gene level language model or something. So it can pretty kind of easily incorporate any arbitrary alphabet, which is a nice feature.
Okay, fantastic. I think we'll stop it there for the sake of time. We landed right at the end of this session and the start of the poster session for Translational Biology and Machine Learning Part 2. Idong and I thank you very much for attending this session. And please, there will be a slide that shows up on how to join GatherTown. It's a little frustrating to punch that in. So in the link here, if you'd like to join the poster session, please connect on the GatherTown. Poster presenters will be there over the next hour, and then we'll conclude with the final session, Machine Learning Part 2, the sequel, in the afternoon. Thanks very much, everybody. Take care.